Welcome back. We will continue the morning's discussions where we talked much about patient engagement and so in the cancer care and cancer research. And we will talk more about prevention and early detection. And all of these questions demands new leadership in order to implement all new innovations and so to get some inspiration on that note, I would like to welcome Roberto Verganti, who is Professor of Leadership and Innovation at Stockholm School of Economics and Harvard Business School. You are with us from Italy, and warmly welcome. Looking so much forward to hear your inspiration.
Good morning. I'm in Sweden actually in this moment, and the first thing I want to say, I'm very, very sorry for the mess created this morning. I really apologize, which means the only thing I can promise that each one of you will knock on my door in the next 3 months. We'll have an Italian espresso and a fantastic croissant here in front of the school where there is a fantastic Italia cafe. And this is probably because I really like to meet you personally when you will be back to Sweden. I will not take too much time. I also apologize for the speakers who come later because I took their time. And I wanted to share some of the reflection about how what is happening in the world in this moment is impacting innovation and leadership. And of course, innovation and leadership in general, but also in the world of healthcare. And I will start from my own experience in the sense that as Ingrid was saying before, I am from Italy in reality. And in 2020, when Italy realized that the pandemic was started to explode, my children are still in Italy. So I had to make a hard choice whether to stay in Sweden. And at that moment, no one knew what was going to happen, or go back to Italy. And Italy, in my case, meant Brescia. Brescia is a small city next to Bergamo. So the choice was going into hell, basically, because in that moment we didn't know what was going on there, or stay safe in Sweden. Of course, I took the last flight with Ryanair before they shut down all the airports, really the last flight, they landed at 10 in the night. And I locked down myself for a few months into the house with the children and my family. And at that moment, of course, as a scholar, I started to receive messages from people and everyone was concerned. And my colleagues and leaders contacted me and say, what is going to be ahead? How will the new normal be? And as a scholar, that was a big question. In the lockdown, there was also a lot of time to think. So I started to reflect, but this was a question that was very, very difficult to address. Just to make an example, this diagram shows the numbers of jobless claims per week in the US. And you see what happened during the recession in 2008, 2009. And then you see what happened in the few weeks of the end of March of 2020. So what happened and what is still happening is something that never happened before in humanity. It's an event that is totally new in the history of what, at least in our life, maybe our parents experienced something similar during the Second World War. But what's happening now is no antecedent, which means that the change we're in front of us is not only new, it's also rapid. So there is no time to build experience. It is humongous. The size of the change is amazing. So the answer is, unfortunately, no one knows. And whenever you have consultants or experts that tell they know where the future would be, in this moment, honestly, it sounds a little bit ridiculous. No one knows how the new normal will be. And when no one knows, the only thing we can do in front of this change is to innovate and change the way we lead and the way we learn and the way we practice leadership. There's no other option. So what I wanted to share with you is no one knows what will happen, but at least we can reflect on what is the way to know and to get to know. So what I would like to do is to share what we thought about innovation, the way you innovate, and some reflection that comes from the research and the dialogue that I had with leaders in this period about the way ahead. So it's not what will happen, but how do we need to change the way we learn and the way we know it? How do we need to change the way we'll discover what will happen? Because we cannot use the same way we used in the past. So 3 things. First, when you are in front of a change of this magnitude, expertise doesn't matter anymore, because what we have ahead of us is totally different than what we met in the past. We know it from the space of healthcare, where the new vaccine, the mRNA, I mean, is based on technology that were actually almost the inventor, the founder of BioNTech was mocked in the industry because no one believed in what they were doing. So the expertise, what we knew in the past, is totally useless to address a scenario that is new. The only thing we can do is simply try and learn. This is very important, and is also very painful for us. I come from universities, and universities are considered to be the centers of expertise. We build our reputation of our expertise. Well, this is the moment in which expertise matters less. What matters more is our approach, is our strategy, is our heuristics, and how we try, how we do experiments, and how we learn. The second change is in the past few, I would say 10 years, the myth of innovation and how do you try, how do you learn in innovation is that you basically do it alone. I mean, the myth of the last few years is, the disruptors, those who come into an industry with a new perspective and they totally disrupt an industry. So we have been praised, the disruptors, those who are good in disrupting competition. Well, in this moment, definitely we don't need disruptors at all. This is a moment in which if everyone try to do it alone, the speed of learning will be too slow. If we want to learn, and we need to learn as the world changes, and it keeps changing as fast that every time we try something, we discover something new. It's too difficult if we try to do it alone, the speed and energy that we have is lower than the speed of change that we have in front of us. So the only way in this moment to learn is to share energies and collaborate. It's what I call learning by sharing. And this is very important. I mean, I work in the School of Economics. I don't mean that we need to share everything, share profits. No. We need to share the learning process. Then everyone is free to compete and is free to have their own ideas. But the learning from the experiments we do needs to be shared. Because that's the only way that enables the entire community and the entire world to grow. I mean, this is the moment in which our focus shouldn't be who has the largest share of the pie, but our focus is how to make the pie larger. So I hope, and we at the university would be the first one to say, being a disruptor is definitely not great. Being someone who creates innovation by sharing the learning and helping also others to experiment, that's a good leadership behavior. The third thing that I wanted to say is that the kind of innovation also is changing significantly. We usually think, I've been teaching innovation for a long time, and typically we think about innovation as a problem-solving process. What does it mean? You have a problem in front of you, and you try to solve it. How do you fix? You have a sickness, and how do you fix it? What I mean is that the kind of innovation that we have in front of us is not simply that we have a problem and we solve it, because the problem we know are all problems from the past. In this moment, if we focus on the problem we know, we tend to solve problem that doesn't exist anymore. What we need to do in this moment is to change the problem we solve. We need to address new, meaningful problems. And I make an example. I mean, first of all, what I'm trying to say is that when we have an innovation in front of us, there are always 2 levels. One is we know the why we are doing things. We know the problem that we know to address, and we try to solve it better. I'm an engineer by education, and engineers are like this. Give me a problem, and I will try to solve it at best. How to make a car that is safer, because we believe the cars need to be safer, how to make a car that uses less energy, and so on. But there is another level of innovation in any space, in every industry, which is the problem that we are addressing meaningful? Does it make sense? And that's is the meaning of what we are doing. Is a change of direction. It's not going in the same direction, even better, but it's a change of direction. I make an example from the space of healthcare, where we work a few years ago. We were cooperating with a company in the space of pharmacies, and that's the classic pharmacy that you see around, and you see here, what is a pharmacy? What is the meaning of a pharmacy? A pharmacy is a supermarket where people who is sick go to treat and try to be healthier. You see down there are the pharmacists behind the table, a little higher because they are the experts, and they are separated, of course, from the patient. A few years ago, we cooperated with an Italian company called Aboca, or Apoteca Natura. It's one of the largest company in the space of her medicine based on natural ingredients. And they wanted to rethink what is a pharmacy in a world. And I hear the word before from Ingrid, in a world where healthcare is a lot about prevention. I mean, if healthcare is about prevention, first of all, people will get into a pharmacy when they feel good, not when they feel bad. So the meaning of a pharmacy is not a place where you go when you feel sick. The meaning of a pharmacy is a place where you go when you feel good, because you don't want to get sick. Second, there's no place in the world where you can get good scientific advice or how to stay healthy. You go to a doctor when you're sick, but who should I talk to when I'm healthy and I want to stay healthy? So they totally redesigned the pharmacy, taking off a lot of the products, which was a very brave approach, and putting what they say the concilio, the suggestion, a dialogue between the pharmacist and the patient, which is not a patient, it's a person who feels good. So you see here in the middle, there's no barrier. The pharmacist is outside of the desk, talking to the people, try to understand what should I do to keep feeling good. And in cancer, we know that it's a lot about a big change of meaning, and it's that's not a technical scientific revolution. It's a meaning revolution, which is first of all a change of direction, and if we want to bring treatments into prevention, we need to think not in terms of healthcare, but in terms of life care, which is a totally different perspective. These are two different ways of change. So what I mean here is that innovation is increasingly moving, not only, of course, we still need to find solutions and still believe that engineers are relevant, but increasingly innovation and leadership is not a matter of finding talented engineers, because talent, expertise doesn't mean anything, and engineers are important, of course. But what is more difficult nowadays is to really change direction in what we do. So innovation is becoming increasingly a job and a task for collaborative leaders. It's about trying, collaborating, and a work of leaders. So that's the reason why I think that the Zero Cancer Initiative is fantastic, because it's really a work of collaborative leaders together. So you are anticipating not only what is relevant for cancer, but what is a good practice for the world of management in general. So there is a lot to learn from what you're doing. Thank you very much.
Thank you so much, Roberto Verganti, for sharing your thoughts with us. Unfortunately, we don't have so much time today to ask you much more questions, but we will bring your thoughts back into the panel here later. Thank you so much.
And you're always welcome to a coffee here, so you can ask me questions where I owe a coffee to every single participant.
Good. Thank you. Bye. And we will then dive a bit more into prevention and early detection of cancer. And I would like to welcome our first keynote speaker on this subject, which is Beatrice Melin, who is a professor in oncology at Umeå University in Sweden. And you will give us a keynote on saving lives through sustainable cancer prevention and early detection. Please welcome.
Thank you very much for this nice introduction. So I'm going to give you a little bit of my view. I work as a professor in oncology, but just as many of you talked about, I met a lot of very sick patients, and that has led me to working with how do we really find cancer early, because that's one of the important game changers that we need to really do, not only talk about, but do in close future. So I'm trying to move my slides here. So why should we find cancer early? I think most of you know this, but it's also a matter of looking into the citizen and the patient's perspective. In the patient's perspective, we would find smaller tumors that would require less treatment and better survival. In the healthcare perspective, we would have less patients with metastatic disease to take care of, which would be good for us. In society perspective, it's really a lower cost of care, better quality of life. It would require a lot of citizen engagement. I think that's where we are right now. We need all individuals to really take their grasp of what can I do so that my cancer is not detected too late. And I think there's a mix of both targeted and broad approaches that are necessary to really have an impact on early detection. What you have to remember is the etiology of cancer is diverse. People often think about it that it's a very environmental disease, and partly is, but it's also complex genetics. And we must remember, cancer is also due to chance. So for many patients, it's not that if I have done this or that, that meaning giving guilt to the patients. We shouldn't do that because in many cases, it's just pure chance. And I think what you have to bear in mind here is it's a complex perspective that we have to have, not only looking at biomarkers of early detection that I'm working with in blood samples, for instance, but also to take into account the full patient history regarding lifestyle, income, access to healthcare, exposures, cultures, and so forth. And this was brought up very nicely in a comment in Nature last September. How could we, if we then had a blood sample test to detect cancer, which is where I'm aiming at, how could we use it? This is a review from Sophia Harled and co-workers from Umeå University, and it's actually, we could use it in many different ways. In lifestyle changes, if we have biofeedback that helps us monitor how we actually improve our lifestyle, that could be of help. We could use it in pharmacoprevention. We could use it at early diagnostics as part of a colorectal cancer screening project, for instance. And we could also use it in clinical diagnostics when a patient has specific symptoms in the general care pathways that we have in the Nordic countries, for instance. And thinking about this, so if we did a blood test for early detection of cancer, where could we save most lives? I think we should think about this carefully, and therefore I made a sort of inverse list, not only bringing up the big fives, but the cancers that really are causing the most death. And here you can see that lung cancer comes first, colorectal cancer comes second. In some statistics, they separate colon and rectal, and then they drop, but really it's the same organ, it's the same disease, so it's second. Prostate, pancreatic, breast, and so forth. And there are the European Code Against Cancer with 12 ways to reduce your cancer risks that is evidence-based, that we all could work towards. And the picture here is just showing colorectal cancer in how you survive at stage 1. This is a little bit old slide, but still you could see my point. The gain we could have from finding stage 1 instead of stage 4. So that's what we are aiming for. Let's see. So the question is, of course, then, okay, we could try to make a blood test to find cancer early, but could we really find cancer at the very early stage? And I think we need to balance the fact that, for instance, when you do general screening, you have to balance that there is a risk of overdiagnosis. So for most of these, what I think will be combined risk scores of different biomarkers in the future, you have to balance and weigh so that you don't get too much overdiagnosis. So I would say the jury is still out. Are we able to find the stage 1 cancers by a blood test? The blood test that we have available is still too imprecise, but still we have tests like PSA and we are using it. So I think that everybody thinks about it like it would be the perfect test, but you know, it may be that the test is not perfect, but we will still be able to use it. And so there are many potential biomarkers, all different types of aspects. Sorry, I was dropped out. I hope you hear me again. Do you hear me again? Yes. I'm continuing. So there's a lot of different biomarkers ongoing, like broad proteomic platforms like Olink and SomaLogic, transcriptomics, and metabolites that could be measured in a broad way, and with small volumes, which is important if you have spared plasma volumes that you want to find out how good are these. You often have a lack of volume in your samples. And I think in the future, there will be a combination of these that makes us really find a blood test for cancer. And one test that I find really interesting and is a test in pipeline by the Grail company is finding DNA methylation parts. They are tissue specific, and that means that you take one blood test and you can really find a lot of different cancers. That is unique to this test. And it's not only that company, but also other parts of the world. People are seeing similar results. So I really think it's beneficial. And there are large studies ongoing in US and in UK. But always you have to understand it's not one size fits all. I don't think it's useful for breast and prostate cancer, for instance, as in terms of the studies that have been published so far. And the sensitivity for stage 1 is really moderate, but you can find a lot of cancers in stage 2 and 3. And also notable, some are positive, but you really can't find the cancer. And I think that is not so strange that happens, but you have to, of course, to have a procedure. How do you take care of these patients that you get a signal that should be a cancer, but you can't find it? In Umeå University in northern Sweden and region Västerbotten, we are working on a study that is called PREDICT. We have collected blood samples since 1990 in several different studies. And now we have around 200,000 blood samples collected. And then over time, and people are giving the blood samples as general health checkups at 40, 50, 60. And now we really have a very long follow-up, and people have got different types of cancers over time. And now we are doing and transforming this to find biomarkers for early detection of cancer, but also for other diseases. So I don't know if you lost me now, but I'm continuing my presentation. I think I shut off my camera and see if it helps. So
we both hear and see you, so that's fine.
No, I think the connection is a little lagging, so I put off my camera. So the PREDICT cohort is going to be around 30,000 cases of different types of cancer, of different types of diseases. Half of them are cancer. We are giving an invitation letter to 74,000 people, and we will have the same cohort controls. They have given blood samples at 40, 50, and 60, and they have been saved. The plasma have been saved in -80. So it's really a unique resource. And there we are trying to understand the trajectory of disease and find disease early. So we hope there to make our contribution, because we, of course, understand that there's a lot of initiatives going on, but we see trajectories of both metabolic and proteomic by the large, broad platforms that we're doing. We can see combinations of biomarkers that seems to observe disease early. So turning towards the citizen engagement in this, what we do in the PREDICT study is that we ask the patients and individuals if we find something clinically relevant, because if you do, for instance, genetics, we might find hereditary cancer genes, for instance. Do you want to know it clinically? So we have a direct contact with our PREDICT participants. We also ask them, if there is a new blood test that we want to do a clinical trial for, do you want to be recontacted? Because we might see some risk profile within your blood samples that make you eligible. And then people say yes and no to participation. So in that sense, we're doing a new platform for precision health in northern Sweden. And I think beyond just finding this magic blood sample test, it's really some important components in the work for early detection of cancer. And what I want to show with this slide is that actually patients, where are they? They are at home, and mostly they don't consider them being a patient. They are a citizen, right? And then you have different types of disease and you should live with them. The time you spend at the university hospital is really minor, and you should have both good knowledge among the citizens to be able to, be at home as much as possible, to have good digitalization, of course, to have novel approaches to blood testing, like taking the blood test at home, not having to go to the health center or the pharmacy. And what we need to remember is that there are many promising biomarkers, but there's really a long way from discovery to the clinic. And there, I think Roberti pointed out very well that you need to share. You need to share what you're doing. You need to bring in new ideas of what's going on so that you move the field forward, because the time of working in silos are, that's the end of this and the beginning of something new. And with this, I was going to stop sharing my slides and say thank you.
And thank you so much, Professor Beatrice Melin, for this perspective and facts that we will discuss further a little bit later on. We will get two more projects described here before the panel discussion that really showcase the progress being done within prediction and screening technologies. And first, I would like to say welcome to Henrik Grönberg. So we turn to Stockholm again, and welcome to the stage, so to say. And you are a professor in cancer epidemiology at Karolinska Institute, and you will tell us more about how to diagnose prostate cancer.
Yes, thank you very much, and for inviting me to this very interesting day. So what I'm going to do in a very short time is talking about how to detect prostate cancer in the best way. I'm going to talk about a new test that we developed here at Karolinska Institutet. Those are not my slides. That's right. A new test that we developed here at Karolinska Institute, it's called Stockholm 3. So, as Professor Melin told you about that, prostate cancer is a very common disease cancer and is one of the big five, as she called it. And this is actually the global numbers of prostate cancer looking 2020 and estimated for 2030. And today it's about 1.4 million men are diagnosed with prostate cancer. It's almost as common as lung cancer. Fortunately, it's not as deadly as lung cancer, but still there is a lot of men dying of prostate cancer worldwide. And just expected 10 years from now, the numbers will grow almost at 2 million new cases a year worldwide. So this is a really big challenge you have to tackle prostate cancer in a good way. And the numbers here on the right, you can see the numbers of 1 out of 8 diagnosed with prostate cancer in the lifetime. That's a US number. And 1 out of 40 will likely die of prostate cancer. But one very important thing is if you diagnose it early, as Professor Melin alluded to, the survival rate, if you diagnose it early and in a local stage, it's almost 100% at 5-year rate. The problem with prostate cancer is that there is no symptoms and signs before it's too late. So you have to diagnose it in an asymptomatic stage. We've had the PSA test, and I think most of you have actually heard about the PSA test. PSA is a blood-based biomarker. It's a protein that is secreted out of the prostate. It's been used now for 35 years to detect prostate cancer earlier. The problem with PSA as a biomarker is that the specificity and the sensitivity is quite poor. It's still one of the best biomarkers in cancer today, but still the sensitivity, you probably lose 30 to 50% of clinical significant cancers, and 70 to 80% of men with increased PSA do not have clinical significant prostate cancer. So you have to improve this. So the way we've been working is that we said that why don't we combine many things into one test? And as Professor Melin said, that you can combine proteins, you can combine genetics and clinical data into one test. So that's what we did in the Stockholm 3 test. We took 5 protein biomarkers that are associated with clinical significant prostate cancer, about 100 genetic markers that is associated with the heritable component of prostate cancer, and clinical data, risk factors for prostate cancer. Put it into one algorithm that tells you if you have a high risk, a normal risk, or low risk of having clinical significant prostate cancer. We started this work in about 10 years ago with a large study, the Stockholm 3 study, where we invited over 58,000 men in a large clinical trial. And then we have published over 25 publications since then. This test is now in clinical use, both in Sweden, Norway, and also now in Spain and Italy. And over 30,000 men have been using this as a clinical test so far. So we've been really working hardly to get really good scientific and clinical evidence that this is actually improving the diagnostics of prostate cancer. So this is actually 2 examples that we published of where we have been using the Stockholm 3 test in routine clinical care. We've been using it firstly at Capio St. Görans Hospital in Stockholm. And then at the left, you can see that we use this to detect. We used the Stockholm 3 test. Then we used an MRI camera. And then we used what we call targeted biopsies, that we can use the image from the MRI to look directly into the and actually take the samples exactly where it's most likely that the cancer is growing. And the data from Capio St. Göran, we've been using now this for 4 years, is you can see that we can reduce the overdiagnosis. And as the last speaker told you about overdiagnostics, overtreatment of insignificant cancer, we've been cutting that in half compared to the rest of Sweden. We detect 70% of the cases that we detect are in the localized setting, and very few in the more advanced setting. So we can show, compared to the rest of Sweden, that we, with this diagnostic chain, have a really good result. But in addition to this, we also look at health economics, because that's very important when you introduce new things, that we actually can reduce the cost of diagnosing and treating prostate cancer with 17%. The second example is on the right. It's from Helse Stavanger in Norway. They said in 2019 that they just switched the PSA in about 100 healthcare regions, or healthcare primary healthcare clinics in the Stavanger region. They switched from PSA to Stockholm 3, and they've been using MRI already for at least 5 years. So this is the result when you just switch from one blood sample to another, and you can see that over a 6-month period, just switching the blood sample, you increase the number of clinical significant cancer almost by the double, and you decrease the number of low-risk cancers. But also here, the health economics show that you can reduce the cost of diagnosing and treating prostate cancer by about 25%. So these are really good clinical examples of real-world data. The last example I'm going to just show you is a study that we published this year, or last year in 2021, in the Lancet Oncology and New England Journal of Medicine. It was a randomized screening study. This is screening by invitation. You invite men at the age of 50 to 74 to a screening program. And in this study, we used both PSA and the Stockholm 3 test. And then we randomized to either systematic biopsies without MRI or targeted biopsies with MRI based on the blood tests. And the summary of the results is that compared to PSA, which has been the standard of prostate cancer screening in the world, you can actually reduce the number of unnecessary biopsies by 76%, which is really amazing. But you can also increase the sensitivity and find more clinical significant cancers. And compared to use PSA with MRI, compare Stockholm 3 with MRI with PSA and MRI, you can reduce the number of MRIs in the screening program in almost 60%, and the reduction of overdiagnosis of small insignificant cancer with 39%. And I think with this study, we've been really showing that now we have the tools to diagnose prostate cancer early in a very efficient way, to combining a blood test, Stockholm 3, with MRI and targeted biopsies. And I think the publication of these two papers have led to a lot of interest worldwide. And I think we will have a revelation when it comes to evaluating screening programs for prostate cancer. And at least in my view, this will have, will have a really large impact on the mortality of prostate cancer. So that said, those were my 6-7 minutes that I was allowed. So thank you very much for listening.
Thank you so much, Professor Henrik Granberg. A very impressive work and a clear example of what is possible to do if we just combine and think new. We will go from Stockholm to Hong Kong. I will welcome Dr. David Lam, who is Clinical Associate Professor at Department of Medicine at the University of Hong Kong. And you will allude more of the subject LDCT screening for lung cancer, the next big opportunity in cancer detection. Please, welcome.
Thank you very much for inviting me. And thank you very much for the organizing committee and the Vision Zero Cancer for inviting me to present this. And as Professor Fagani has mentioned before, it's very important to have this opportunity to learn and to share from you and also other colleagues in the field. And what I'll be sharing with you is low-dose CT scan screening for lung cancer. And that represents actually a big opportunity for cancer detection. So, but first, what is low-dose CT thorax? And indeed, this is how it looks like when we use it in the clinic, that it is actually a more detailed scan of the thorax with slices of the lungs that you can see that actually is a computer reconstruction of the lung images in slices. And talking about lung cancer, you can see from the screen that if we put the prevalence of lung cancer in the world, especially in men and women side by side, you can see the lung cancer actually like two different diseases.
Sorry to interrupt you. You have to switch slides from your side.
I'm switching. Yeah.
Oh, you are.
Is
Sorry.
that all right?
That doesn't. We can't see that here. So, are you in presenting mode?
Yes.
Now we see that you switch.
Oh, you see the slides. Oh, sorry. So if I can go back to the last slide, that's what I was talking about. That's the appearance of the low-dose CT scan that you can see. It actually represents a type of radiological imaging that is a computer reconstruction of the images of the lungs being cut into slices. And you can see the presentation of the lungs actually presenting slide by slice. And that's what we called low-dose CT scan as a tool for lung screening. And if you look at the current prevalence of lung cancer in the world, that's actually, if you look at the prevalence of lung cancer of men and women being put side by side, it appears there are different diseases of different preferences. As you can see, the prevalence on the left-hand side of the screen and the women's prevalence of lung cancer on the right-hand side of the screen. And but if you try to put the prevalence of smoking side by side, the prevalence of lung cancer in the world, you can see the distribution of lung cancer is quite similar to that of smoking. And that means smoking, mainly in the man, is more or less a disease quite related to smoking, but not the woman. So that's why if we try to project the lung cancer prevalence or the burden by means of smoking, you can see with the world's smoking prevalence going down a little bit in the coming years, projected to be, actually there will be more and more non-smokers with lung cancer to come up, although we are not saying that smokers are not important, but we are trying to say that actually when we talk about lung cancer screening, we are dealing with quite different populations in the men and the women, the smokers versus the non-smokers. And you can see, especially in this part of the world in Asia, there is a rising trend of non-smokers with lung cancer compared with other parts of the world like Europe and the United States. And so that's why it's getting more and more important that we need to tackle lung cancer screening in different ways. And unfortunately, without screening in the past, we believe that lung cancer is actually a lot of the time diagnosed at an advanced stage and represents a poor prognosis because it cannot be resected. So lung screening has been done in the past, mainly on smokers. As you can see, the NLST study and also the Nelson's trial that's held in Europe actually have shown benefits of smoking, of lung cancer screening when we try to target the appropriate populations, that is heavy smokers. But as mentioned, what about non-smokers? And we believe that from previous experience of screening, even in smokers, we have the projections that successful screening will actually means that there will be a stage shift of diagnosed cancer from late stage to early stage. And so that represents early cancer detection. And with screening trials being held in different countries, different regions, actually, we also identified, besides smoking and age, we are able to identify other risk factors that are good for prediction of the risk of lung cancer. But unfortunately, with screening technology, even with low-dose CT, there are a lot of byproducts that come up from the screening, and that's what we call indeterminate lung nodules. As you can see from the screen, that CT scan are able to pick up lung nodules or lung masses of different size. Not all of them are lung cancer, but of course, by size, a lot of the times the bigger ones are the definite lung cancer. But how about those small size, like those on the left-hand side of the screen, those small nodules, especially sometimes that could represent scars or reaction to previous tuberculosis. So we have a lot of these problems actually got detected alongside lung screening program. And we are devising different programs or different treatment algorithms to manage these pulmonary nodules incidentally detected. And it's important to follow these lung nodules up because they have the potential to become lung cancer in the future. As shown in this diagram, that actually the screen, the scan on the left-hand side actually initially represent the patient got detected with small lung nodules on initial lung screening, but with subsequent follow-up, actually it becomes a lung cancer of adequate size that actually, unfortunately, by that time, become unresectable. So it's now more trendy initially, indeed, to have a lung screening program, to have a lung nodule program to run side by side in order to catch patients who are not initially eligible of lung screening, but at the same time, they have incidental finding of lung nodules that the follow-up of those will be able to be an opportunity for us to get lung cancer detected in the early stage. So that's why the summary, if I can, for today's sharing with colleagues is that an incidental lung nodule program will be able to promote the diagnosis of early stage lung cancer, and that would, if run side by side with a lung screening program with low-dose CT as a tool, and also appropriate risk prediction modeling, that we're able to improve patients' outcomes. There are many issues that we are facing. For example, what's the early time, what's the appropriate time for appropriate investigation and follow-up, false negative and false positive, and at the same time, how about those biomarkers or even AI could help us to improve this LDCT and also the relative risk prediction program for screening. That remains the future research questions to address. And so sorry for the initial issue with the presentation, and here I'd like to thank you for your attention, and I would welcome any questions or panel discussion later. Thank you very much. If I can stop sharing here.
Thank you so much, David, and we are very glad that we got everything to work in the end. And now I would like to welcome our panel up on stage here, which there is then Hans Hägglund, Her Excellency Omniat Al-Hayeri, Humaid Al-Shamsi, and Andrea Balaghi. Please welcome.
Thank you.
دكتور السلام عليكم.
السلام عليكم.
كيف حالك؟
So I'm curious to, now we have heard a lot of different examples here on early detection and what is possible to do. So I want to turn to both Her Excellency and Humaid here to begin with. how are you working with early detection and methods like this here in the Emirates?
Thank you for the opportunity and thank you for the invitation. It's a topic that's very dear to my heart. Dr. Ahmed is the real expert on the ground, but I come into this from a public health perspective and also as a daughter of a survivor of breast cancer. So to me, I think it's all about my own 3 Ps and an I, and I'll tell you what is that about. So it's prevention, personalization, proximity, and then the I for me stands for innovation and artificial intelligence. There is a lot that's already have been done on the ground in the Emirate of Abu Dhabi and in UAE in general. So we do have the screening blocks in place. We were trying over the last 10 years to bring those forward, improve them, enable people to access them as soon as possible. So we've recently put all our screening programs, including cardiovascular risk screening, which are also contributing to the risk of cancer, and lump them together with packages that are age appropriate. So cancer screening now is actually provided by highly qualified providers that have very high standard, have multiple units that are accessible to people, as in primary healthcare setting, so it's close to where they are, and at the same time they have the follow-up, and frankly they cannot be paid unless they do your full package. So we are trying to close the loop. Yet I think there is so much that we need to bring into this. For us, it's not a marathon. It's more like a relay race. We have to work on that as a community. We have to advocate. We have to build strong partnerships. We have to learn when to give the right piece to the best player, so that we ensure that we all reach the target that we want to reach to. So within the prevention, we are looking for improving the detection as early as possible. So we are looking into child wellness visits, ensuring that people are being followed all through their life cycle, so that if there is any sign of changes that could be related to cancer, this can be detected early on by a physician that has been going through the life cycle with them. So it goes from child wellness visits to comprehensive school screening to our adult screening IFHAS that has been launched recently, which covers all the components that I've talked about. For personalization, we really look into integration, integrating our work with our colleagues in the UAE Genome Project. So we really want to personalize it as early as possible to look into the multiple omics in general, so genomics, proteomics, fragmentomics, and all the omics that the colleagues there know much better than me. We want to bring all of that together to the service of our population, locals and residents within Abu Dhabi, to ensure that we pick up as soon as possible what needs to be done, and at the same time have a personalized plan. Meanwhile, we also have under our umbrella and public health, the mental health and the ethical considerations. So we have to ensure that we don't overdo it, or label people very young in their life and keep them with that label without giving them enough genetic counseling and support, without giving them enough mental health support, without giving them social support. And that's all of the bigger picture. Proximity, again, younger age, closer to your place. Link that to innovation and artificial intelligence. If we can deliver the test to your house, we want to invest on that and ensure that that's the preferred choice if it's accurate enough. And we want to utilize artificial intelligence opportunities. So one of the main reasons for us joining colleagues like you is to be introduced to the new innovations. For example, the utilization of artificial intelligence for early detection of lung cancer, opportunistically using our visa screening, normal X-rays. That's an amazing initiative that we'll be working on hopefully soon with our colleagues, and in collaboration with the Center of Innovation and Research within the DOH. So Abu Dhabi Public Health Center takes this as a legacy, and we try to fit all the pieces of the puzzles, deal with our colleagues and champions within the sector, and ensure that we together make the difference.
So Dr. Hamid, do you feel that you, with all the technologies available, are they coming to the patients here?
Okay. So I cannot agree more with Dr. Omniat Al-Hajri about her comments. I think the first statement I would like to make, to reach the Vision Zero cancer, it has to go through early detection. If we talk about the current treatment, immunotherapy, targeted therapy, we are trying to achieve 10 to 20%, control of cancer in advanced stage. We don't want to reach that stage. We want to be able to detect cancer very early so we can save lives. I think one of the biggest issues here in UAE is the culture. I'll give you an example. We're talking about, the early detection using blood samples. We introduced the first, blood-based sample for colon cancer using a test called imiprocolon, which is an FDA approved. The uptake is very slow. The culture is not there. We need to work very hard to try to persuade people to come to talk about cancer, to be open about cancer, and also to do the test. If we talk about colonoscopy, forget about it. They're not going to do it. The CT scan, even it's done in Abu Dhabi, the uptake is very small. If we look at the actual numbers of the screening of cancer in general, breast cancer, cervical cancer, they're very low. I'm not going to disclose the number, but they're actually very disappointing. Maybe not in Abu Dhabi, but in the rest of the Dubai and also the rest of the Emirates, they are less than around sometimes 10%. If you have a very effective screening, it has to be up to 50% and above. So the culture is not there. We need to work very hard to change the culture, the mindset of people to accept to talk about cancer. Now we're talking about cancer here in this country more than what we did like 5 years ago. The culture 5 years ago was more like in the 60s and 70s in the US and the West. People, they used to call it the disease. They even avoid mentioning the word cancer. But now we are more open. So I think it will take time to reach the target. But certainly also the other thing I would like to mention is we need to have a call and a recall system in place. Currently, we don't have this. As Dr. Omniat mentioned, it's just, based on the patient presentation to the GP. Oh, maybe I need to do a screening for you, but there's no structure where we call the patient, we send them a reminder, an email, a letter, a phone call, so they come and do their test. So I think targeting the call and recall system and also changing the culture are very, very critical before we move forward with all the fancy tests. I think we need to get our basics first, and then we can move with the technologies.
That's really interesting because it's we did discuss or led the discussion with patient engagement and so earlier today, but it's quite easy, I think, in the healthcare sector and researchers and so that the focus is that we can cure and we will cure, and it's always focused on the improvements and the technologies, but. Of course, especially with early detection, there's a lot of ethical questions, and Beatrice Melin were addressing some of them. How do you, because if the culture is that you don't really even want to test yourself with genetics, for example, the result isn't just your own. It may be relevant for your siblings, and you haven't asked them. And so, how do you feel that in implementing new technologies, how to bring with the ethics through the well? Hans, do you have any?
Hello.
Yes, you are with us.
That's a tricky question, I think. But to start with, I would say that prevention is the best medicine. And the next best is early detection. And when we talk about early detection, we talk about trying to find new cancers. But we cannot forget, early detection for relapses or second cancer. So both in the European Cancer Plan, the Swedish and other countries' cancer plan, this is this is stressed that we have to work with this, and I think that we in Sweden try to use different standardized care pathways, national guidelines to help the primary healthcare to find cancer early on. But what Beatrice Melin said, there is also a citizen engagement here that we have to teach and learn ourselves. When do we need to contact the healthcare? Because what happens before we go to the primary healthcare, where 70, 80, 90% are detected, it could be 1 week with symptoms, a month, or years with symptoms. So I think we should focus on also the citizen engagement to learn and educate people? Well, coming to the ethics, I think that Beatrice touched upon this, and when they asked the question to those people or the citizens that are in that study, the majority said, I would like to be contacted. And the relatives also said, well, I also would like to be contacted if you find something.
And how do you handle the ethical questions?
Let me tell you about a short journey that we've had. So we've started our screening programs in 2007 nearly. We've learned a lot during that period. So what's happening for us now is we're being very careful whenever we release a new initiative and to address the concerns. So for example, the revamp of our comprehensive screening like FHAS have included a call-recall system that will hopefully bridge the gap in the Emirate of Abu Dhabi. It also have included the mental health screening element, which is an introduction to the discussion about what will happen when the results of your test actually come, and it will give us a way through to open with the patients. Now, the main reason that we still did not launch the connection to the genomic is the fact that we still need to build our capacities, have enough genetic counselors, have the right rules and regulations that controls the tests, the outcomes, and what happens after that. So we are taking things very carefully from a regulator point of view, and within the Abu Dhabi Public Health Center, we try to ensure that we do not overpromise. So you don't open something to people and then tell them, I don't know where to take you next. And we are sure that by the time we do things, we actually test them before. So this is our approach. So FHAS has been launched now from the beginning of 2021, but it's still in a pilot phase. By the end of the first quarter, we are getting the full feedback from our colleagues that are actually on the ground delivering the service to see what needs to be reformed, how can we improve it, can we use databases that we have to contact people to come for the screening, how much is that impinging on their personal rights and choices. So there are so many questions, but we are taking those actively.
And how has the feedback been then? Have you had to change anything in your processes based on the feedback? Or?
We are actually in the process of getting the final feedback.
Okay.
All the feedbacks we got yet were basically on the actual implementation and the difficulties.
Okay.
Because for the provider to be paid, he has to complete the full battery of testing. This is a new pressure that we are putting on the healthcare provider to ensure that they take the extra step to reach their population. And that's a painful step because it makes you wait till you achieve the full target to be receiving it. And I think this is most of the comments that we've got in addition to IT issues. But the full analysis is expected by March. And hopefully it will be discussed with providers and caregivers, and I'm sure there will be multiple solutions.
And I wonder, Andrea Ballag at Olink, you are in the forefront of science and what is possible to do with proteomics and genomics and so on. In your work, how much do you think about the implementation at the end, so to speak?
A lot. I mean, if we would like to make our research impactful, then it would need to be implemented. But we see a lot of sort of challenges with the implementation, and many of those were mentioned here. So one of the things is that when you have these new results coming out from scientific work in academic publications, then very often there is a stop there. So the current system, regulatory system, approval system, at least I would say in Europe and in the US, it's very, very cumbersome, lengthy, and very expensive. So I think there we would need to think differently, and we would need to allow research and scientific achievement much earlier, coming closer to the patient, to the clinic.
And how would that be possible? What can you see as a concrete?
Yeah, a very concrete example. I could mention one example. We developed a proteomic signature that could foresee in secondary prevention patients who had already had a cardiovascular event, what is the risk that they're going to get a secondary event again. if you have a proteomic signature like this, very similar to what you have heard about the prostate cancer screening from Professor Grönberg, then it's a very cumbersome process to start to so-called validate it in clinical use, because the regulatory framework doesn't allow you to start to use something which is research use only. And I would say there are places in the world, Sweden is one of those, where we have this little bit of gray zone that we sort of can try it. But for example, if you think of the US, FDA is very, very sort of rigid in this term, and it's a very expensive and cumbersome process to even evaluate something in close to the patient.
And I was thinking that Beatrice Melin in the beginning mentioned that the bottlenecks are different for different diagnoses, and you mentioned culture here as one of the bottlenecks for getting the screening working. How much can we learn from each other? What is a common ground here that we can learn from each other, or is the challenges very specific regarding on depending on what regulatory system or what culture or what diagnosis? Yes.
Maybe we just missed that. There is one good thing, I think, or positive consequence of the pandemics that we just went through or going through. Everybody suddenly got much more interested in their health, in vaccines, in medications. Everybody became suddenly a vaccine expert, you know. So I think we should build upon this. It's a perfect opportunity to talk about more health, talk about our own individual responsibility for our own health. So maybe
yes,
that's an opening.
Well, I will highlight this based on our research. Based on the genomic testing, we do have some differences between the UAE population and the Western population. We published data in 2016 about colon cancer, and there were some differences. We published another paper 6 months ago about breast cancer, and there are some differences. We also published the differences in the rate of or the incidence of cancer in our population. In UAE, 20% of breast cancer are under the age of 40, and 40% are under the age of 50 in colon cancer. In fact, this morning I saw a patient who was 37 years of age with colon cancer. How can we, and this is a question I get all the time, and I don't have an answer. Doctor, you always tell us to screen at the age of 40 for colon cancer, but we're getting cancer in the age of 30 and less than 40. How can we explain this? I think the current methods of screening are not perfect. I think we're getting more into the liquid biopsies and liquid screening, which will ease the age restrictions to make it more generalized. But I think we still have a lot to achieve, to be honest with you, for our own population when it comes to the age and the onset of the cancers, and also the way to deliver these screenings for our population.
Okay. Hans Englund, you...
Well, I think we have much more in common than differences, but I think that when it comes to organized screening in Sweden, we know that we have difficulties to reach out to certain regions or communities, especially in low socio-economical places. But what I think was interesting from the previous talks was that what we have in common is that the physician's umbilical cord to the patient is hooked, and we have to cut it. And I think that another thing that I heard was that we have to use the cell phones as new hospitals. We cannot build more hospitals, and if we use that, we can find cancer earlier, I think, using the information from the patients.
I have to thank the panel for this, and the discussion will go on during the lunch, I think. You're welcome to keep discussing this, and we will come back after lunch as well and keep the subject alive. So thank you so much for now to the panel and to the audience and to all our speakers, and we will be back here again at 2:30 local time in Dubai, and that is 11:30 in Central European time. Thank you.
I believe that we are in very exciting times in the history of life science. We are now analyzing and characterizing all the building blocks of human beings. The most important building block is the proteins. Traditionally, it has been very hard to do this in a very comprehensive way, but there are now new technologies to do this. And one of these technologies is proximity extension assays. And this allows us now to analyze in a very sensitive and accurate way a lot of the proteins which are present in blood. And that is why I'm so excited about the future.
Olink Explorer will be a game changer for proteomics. This new development combines our elegant PEA technology with a state-of-the-art NGS platform. I find it exciting that we've now reached a higher multiplexing level and that we're also using a semi-automated protocol. And with that, we can achieve increased throughput, but at a lower cost.